So where we're headed now is to pick up where we left off last time. And I basically, you may look at the videos and see that I basically spliced together a bit of the first one and a bit of the second one because I got farther in the first one than the second one. But the second one was better for the most part than the first one, so you get a kind of hybrid of the two. So what we're going to do now is we're going to emphasize this idea of DNA semiconservative replication. We've established the idea of the organization of DNA. We've talked about its components. We've talked about the anti-parallel strands. We've talked about being oriented 3' to 5' on one and 5' to 3' on the other. And now we want to think about how we make new copies of that molecule, because after all, life on the planet requires that it be able to replicate itself, make copies. And so what we're going to be doing now is drilling down into the chemistry of making copies of these molecules. And there are a small number of rules about this. The small number of rules are, first, we have a protein called DNA polymerase. The DNA polymerase can only read the DNA strand in the 3' to 5' direction. It's unidirectional in that fashion. That's going to call into play some rules that we'll have to abide by when we make new copies of DNA. The other thing that the other idea of the DNA polymerase, in addition to its reading orientation, is the idea that it generates base pairs. That is, the base pairing that we discussed last time is now going to be imparted here, that we just made up last time. That base pairing now is going to be derived from the action of the polymerase. It will go out, find the right base, and form the appropriate base pair. Now remember, too, that those purines and pyrimidines can only fit together in one way. And so once you bring those purines and pyrimidines close together in that mechanical fashion, they'll bind to each other by hydrogen bonding, stay there for a long enough period of time so that the DNA polymerase can do its other major job, which is to form the phosphodiester bond. Remember last time we emphasized that these polymers are polymers because the sugars, the deoxyribose sugars, are linked by the phosphodiester bonds between the number 3 and number 5 carbons. So here we'll try to describe how this DNA polymerase does this job. Now once we have generated a new molecule of DNA and we have described the idea of semiconservative replication, where we have one new strand and one old strand in the newly synthesized double-stranded molecule. Once we've talked about that, then we're going to go to RNA. Now, one of the major differences between RNA and DNA is where DNA is always organized in the double strand, RNA is often a single strand. We'll see it folds on itself, but it's still a single strand. The single-strand nature of the RNA is because, in large part, because of the chemistry of ribose. The number 2 carbon on ribose contains a hydroxyl, where in deoxyribose there's only a hydrogen. Because of this great bulky group largely prevents the twisting of the strands one around the other. So these molecules of RNA with ribose itself don't easily form a helix. They basically are dictated to have a linear structure. The other dictate here is in the bases. And again, we're going to see 2 purines and 2 pyrimidines. The 2 purines will be adenine and guanine. And the 2 pyrimidines will be cytosine and one called uracil. Now, what I want to impress upon you when we go through this is that the difference between uracil in RNA and thymine in DNA is precious small. In fact, thymine, which is relegated only to DNA, that thymine has a chemical name 5-methoxyuracil. The only distinction between uracil and thymine is that methyl group. But again, that's important because you put this great big methyl group on this molecule, and that inhibits, in the DNA, that methyl group permits the formation of the helix, where it's inhibited in the RNA. So we'll lay out the idea of the structure of an RNA molecule, and then we'll describe the idea that there are a small number of RNA types: transfer RNA, ribosomal RNA, mRNA, or regulatory RNA. But regardless of the RNA type, all of those RNAs originate through a process called transcription. That is, the sequence of bases in the RNA is dependent upon the sequence of bases in the DNA. And again, we're going to use the hydrogen bonding between bases and the rules of the generation of phosphodiester bonds to talk about transcription. When you hear the term transcription, think immediately RNA to DNA. Replication is DNA to DNA. Transcription is DNA to RNA. And we're going to use the information in the base sequence of DNA to make a complementary RNA molecule. But there are some tricks here. Because remember, the DNA, if you think about a eukaryotic cell and you look at a chromosome, you recognize that that chromosome is one great long molecule of DNA. But within that one great long molecule of DNA will be subregions called genes, maybe several hundred of them. And those genes are going to be responsible for making proteins. So now you have a kind of puzzle. You have what appears to be a continuous sequence of bases, and yet biology manages to take that continuous sequence of bases and find subregions within that continuous sequence. The ability to find subregions called genes within that continuous sequence first starts with the idea of the promoter. The promoter is entirely within the DNA. The promoter is a sequence of bases in the DNA called a consensus sequence. We have a sequence of bases in the DNA which are going to be a kind of recruiting site to which a transcription factor will bind. Promoter is always DNA. Transcription factors are proteins. There may be 13 to as many as 14 or 15 separate proteins in the transcription factor. But the transcription factor is a DNA binding protein which finds the promoter and binds at that site. The binding of the transcription factor to the promoter recruits an RNA polymerase. The name RNA polymerase should ring some bells because, after all, it's going to polymerize an RNA molecule. It's going to read the DNA 3' to 5'. It again is going to form phosphodiester linkages between adjacent sugars. But now, instead of forming phosphodiester bonds between deoxyriboses, it will now form phosphodiester bonds between riboses. And again, though, just like we saw with the DNA polymerase, or we'll see with the DNA polymerase, we'll see that the RNA polymerase also finds the appropriate base that's complementary to the base on the DNA. It will find a cytosine and pair it with a guanine. It will find an adenine and pair it with a thymine. And it will find an adenine and pair it with a uracil. And we'll see how that's different. But now we're generating a new RNA strand that's complementary to the DNA. If we were talking about prokaryotic biology, if we were talking just about prokaryotes, we would find that the RNA strand that's generated by the RNA polymerase is absolutely complementary to the DNA strand. One-for-one base sequence. And then we could look at that base sequence in the RNA, complementary to the DNA, and by recognizing that we use series of 3 bases at a time for each amino acid to code each amino acid, we then could look at the DNA sequence and immediately know what the amino acid sequence was in protein. And they did that for years, and everyone thought this is the way all biology would work. And then they began looking at eukaryotic cell biology, and eukaryotes threw them a real curve because we find eukaryotes can do something really clever. What eukaryotes can do in the nucleus is they can transcribe an RNA strand from a DNA strand in the same way we see here. But once that RNA strand is transcribed, they can then cut that RNA strand and tie it back together in slightly different ways. So that now you have an RNA strand which isn't exactly complementary to the old DNA strand. It's been edited. It's been cut and spliced so that now we have a new RNA strand. It's an edited RNA strand. We'll see that this edited RNA contains exons, which will be sequence of bases that will be expressed as amino acids, and we'll see that there are so-called introns in this editing process that are discarded, and we'll talk about that.